Hey guys, welcome back to One Stop Biology. So in today's video, we are going to start with inheritance of two genes. So guys, we are right now studying the units genetics and the very first chapter of it, which is principles of inheritance and variation. And we have already covered inheritance of one gene. And then in the last video, we covered the different laws of inheritance. So I hope you are able to understand this all these videos. So guys, if you do not please do not forget to like the video and share it with your friends as much as you can. And also, if you have any doubts as such even while watching the video, please do not forget to mention it in the comments. You can even mention if you did like the video. Okay, so let's start off with inheritance of two genes. So the basically the way Mendel experimented with one gene, like we saw the example of tall versus dwarf plant, right? In the same way, he also worked with and crossed pea plants that have different characters, as in like pea plants that differed in, say, two characters. Okay, so how basically he did it is, say for example, like in a pea plant, basically the seeds, there can be a pea plant with seed color to be yellow, and the seed would be round, right? There can also can be a plant with seed color green, and the seed would be wrinkled in shape. Okay, so again, this basically is one characteristic, one character, and this is the other character, which is seed shape. Right. So what Mendel found was that the seeds resulting from crossing of these parents had yellow colored and round shaped seeds. So that means that the yellow color is dominant over green and the round shape is dominant over wrinkled. Now, these results were basically identical to those that he got when he made separate monohybrid cross between yellow and green and round and wrinkled. So when he did monohybrid crossed, he found this, right? So he found that yellow is dominant and round as well is dominant, right? So now let's do one thing. Let's study it with the help of the Punnett square itself, and let's just see few examples. So let's say that we are using the genotypic symbol Y, right, in capital for dominant yellow and small y for recessive green. In the same way, we are using capital R for dominant round and small r for recessive wrinkled. Okay, so these are the genotypes, right? So in the parent generation, what he took was he took a plant with round and yellow seed and another plant with wrinkled and green seed, right? Now, what kind of F1 generation he found? So basically, if you see the gametes that he got, was again both are dominant, so dominant gamete of both. So one round ka gamete and the yellow ka gamete. And then here both recessive, right? So small r and small y, recessive green and recessive wrinkled. Now what he did basically when he crossed these, the first generation that he got was round and yellow as phenotype, right? But if you see the genotype here, it's basically all four gametes, right? So if we see all four gametes, it will be R, R, Y, and Y. And since these are dominant ones, they will show the character, right? So that is why the phenotype is round and yellow. Then what he did is he did selfing between the plant of F1 generation. Now you see what exactly was the founding here. So the F1 generation was this, right? Now he did selfing. Now what you'll have to do is you'll have to see all the combinations of these four gametes, right? So it can be, say, this and this. So the first gamete formed will be this, right? And then you will form this with this. So the second gamete formed will be this. Now the third one will be this and this. Now remember that this we are done with the dominant yellow, right? So now we'll form it with dominant red, round and recessive green. So here what you will have this and this, right? Now the fourth one will be the both the recessive one. So these are the four gametes which were formed, and see all the gametes will basically have one trait of both the characters, whether green or red or round or wrinkled. Sorry, green or yellow or round or wrinkled, right? So you'll see these four here, right? These four and these four here as well. Now what will happen is we'll see the crosses. So if you cross this and this, you will get all capital, right? Now what you'll do is you'll cross this with what? Again this. Tell me, am I wrong? This again with this, right? And what you're getting this. You'll cross this again with this. So basically you're crossing all these with everyone, right? So you'll cross this with this, you'll get this, and here again these two, and you get this. Now here if you see, in every case, since we have at least one dominant round and one dominant yellow, all are round and yellow, right? Now in the second box, this line basically, you will cross all this with this one. So basically you are crossing this with this here, you're crossing this with this here, you're crossing this with this here, and you're crossing this with this here. Again, in the third case, you'll cross this and this, you'll cross this and this, you'll cross this and this, and you'll cross this and this here. Again, in the fourth case, you'll cross all four with this one, and you'll get these results. So if you see again this line, what is happening is again you have at least one dominant round and one dominant yellow, all are round and yellow. Here, if you see, since you're crossing this one and this one, you have dominant yellow, but you do not have dominant round. So the seed is yellow, but it is wrinkled. Here again, it's the same case, right? Now, if you go down, you see, so what is happening is it's just the opposite. Right? So it's just the opposite basically. How? You're getting the R, which is dominant round. So the seeds are round, all three of them. But you are getting the all recessive yellow, right? So it's basically round, but it's sorry, recessive green. So it's all round but green in color. And then if you see this last one, what is happening here is we are crossing the recessive with the recessive one, right? So here you are getting a wrinkled green because it's all recessive gametes. So if you see the phenotype here, what phenotype you are getting is basically round yellow, round green, wrinkled yellow, and wrinkled green. And what are the numbers? So if I have to count it, 1, 2, 3, 4, 5, 6, 7, 8, and 9. So 9 round yellow. Then I have round green, 1, 2, 3. So 3 round green. And then I have 1, 2, 3 wrinkled yellow. And I have one, 3 wrinkled yellow. And then I have one wrinkled, this will be green. So I have one wrinkled green, right? So these are the results of dihybrid cross, where we are crossing two parents with two different pairs of contrasting traits, which is seed color and seed shape, right? So now what we'll do is we'll study some variations of a dihybrid cross as well. So let's see what is the first variation. The first variation is law of independent assortment. So we'll see what exactly does it mean. So in the previous dihybrid cross, what we saw was that the phenotype of round yellow, wrinkled yellow, round green, and wrinkled green was 9 is to 3 is to 3 is to 1, right? Now, this is basically was observed for several pairs of characters. Okay, so the ratio of 9 is to 3 is to 3 is to 1 can be derived as combination series of 3 yellow and 1 green with 3 round and 1 wrinkled, right? So you can say this as 3 yellow and 1 green, right? And then 3 yellow and 1 green, and then 3 round and 1 wrinkled. Right. Now, basically, based upon such observation on dihybrid cross, Mendel proposed a second set of generalization, which we call as basically the Mendel's law of independent assortment. Now, this basic law states that when two pair, listen to it very carefully, when two pair of traits are combined in a hybrid, okay, when two pairs of trait are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. So basically, round and wrinkled, right? So round and wrinkled and yellow and green are two different traits. Now, segregation of this pair is independent of segregation of this pair, right? So the Punnett square basically can be used effectively to understand the independent segregation. So if I tell you that two pair of genes and we have segregation of 50% as the round and 50% as wrinkled. Now this is independent of 50% of yellow and green. Therefore, 50% of wrinkled bearing gamete, right? And 50% of green, right? So basically what we mean here is that there are four genotypes of gametes, right? That we saw. These gametes are these four basically. Here that you see, these four. Now, frequency of basically getting one produced is 25% of total gametes which are produced, right? And none is dependent on the other one, right? It's all equal, right? In the same case, if we talk about this and this, or this and this, basically the gametes to be segregate 50% chances of this and 50% chances of this. In the same way, 50% chances of this and 50% chances of the other one, right? So there is no dependency on the other pair of trait for this to be segregated in a gamete, right? So this basically is the law of independent assortment. Now, the second variation that Mendel discussed was chromosomal theory of inheritance. Now, what exactly is chromosomal theory of inheritance? Yes. So, what happened is that Mendel basically published his work on inheritance of characters in almost 1860s, around 1865. Okay. But what happened is that because of different reasons, it was not recognized by 1900. Now, first reason was basically communication was not as easy in those days because there was no, such widely renowned publications who could publish your work, and people would just read it out and people would come to know that okay, this is happening. Now, the second reason was that his concept of genes, or he used to call it as factors, right, as stable and discrete units that controlled the so what we say is that these are the basically units of inheritance, right? So they control the expression of traits. So his concept of gene or factor, which being the the discrete unit that control the expression of trait, or maybe of the pair of alleles which the gene has, was not accepted. What happened is, right, the third point was that the Mendel's approach of using mathematics to explain biology was very new, and again the biologists said that they do not accept it, right? So what happened is in 1900, three scientists, who three? De Rais, Correns, and Shemak, these three independently, not together, independently rediscovered Mendel's result of inheritance and characters. So these three basically independently rediscovered, right? Now after that, so basically by that time there was advancement of microscopy as well, and scientists were able to carefully observe cell division and all that. So that led to discovery of structure of nucleus and how the cell divides and what are chromosomes. Now what happened is by 1902 chromosome movement during meiosis was worked out, and Walter Sutton and Theodore Boveri, they both noted that behavior of chromosomes was parallel to behavior of genes, and used chromosome movement to explain Mendel's law again. So see that after so many years, two different scientists basically explained Mendel's law, which was not accepted at his time because of certain, say, hindrances in mind barriers of the biologist. That why you are basically, explaining biology with the help of mathematics, that was not acceptable, or his concepts were not acceptable, right? So basically, what they did is that they basically, if you see here, they proved how meiosis, through meiosis, you have chromosomes as well. In meiosis, you have chromosomes and as well as genes, right? Now, two alleles of a gene pair are located on homologous sites, and we call it as homologous chromosomes, right? Now, during anaphase of meiosis one, if you remember it, if you can recall it, during anaphase of meiosis one, two chromosome pairs can align, where they can align at the metaphase plate independently of each other, right? Now, at that particular time, again, Sutton and Boveri argued that pairing and separation of a pair of chromosome would lead to segregation of pair of factors that they carried. But then Sutton united the knowledge of chromosomal segregation with Mendelian principle, and that now is called as chromosomal theory of inheritance. Right? Now, how we can basically understand this? If you compare the chromosomes of four different colors, right? So if you compare here the chromosomes of four different colors, right? What you will see is that in the left column, which is possibility one. Now here we have one long orange and short green. So we have long orange, sorry, we have long orange and short green. And then we have long yellow and short red. Here we have long orange, short red and long yellow and short green chromosome at the same pole, right? So if you see the left column, which is possibility one, orange and green is segregating together. But in the right column, orange is segregating with red one, not the green one, right? Now, basically, what Sutton and Bory argued was that the pairing and separation of a pair of chromosomes would lead to segregation of a pair of factors that they carried. But following this synthesis of ideas, we saw that it basically the same theory, chromosomal theory of inheritance, was again verified by Thomas Hunt Morgan, which led to discovering the basis of for the variation that sexual reproduction produced. So he worked with the fruit flies, which are Drosophila melanogaster, right? And he found out that again different they complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny, so that was used basically. And then there was a clear differentiation of sexes, male and female flies both, right? So he again found out the same thing, that the chromosome assortment is independent, right? So what basically it means is that if you see here, once they are segregating in meiosis, here meiosis 2 is happening, and then we are getting these four different gametes, right? Here in this case, you have the long orange and short green. Here in this case, you have the long orange with short red, not green. So they have basically independent assortment, right, at chromosome level. Now, the third variation that he gave was linkage and recombination. So Morgan again carried out dihybrid cross in Drosophila to study genes that were sex linked. Okay. Now he observed that two genes did not segregate independently of each other, right? And the F2 generation ratio deviated very significantly from the usual one, which was 9 is to 3 is to 3 is to 1. Okay. So what happened is that when two genes in a dihybrid cross were situated on same chromosome, remember, when it was situated in same chromosome, the proportion of parental gene combinations were much higher than non-parental type. I'll read it again. When two genes in a dihybrid cross were situated on same chromosome, proportion of parental gene combination were much higher than the non-parental type of genes. So he said that this is due to physical association or linkage of two genes. What he said? He said that this is due to physical association or linkage of two gene, and he coined the term linkage to describe this kind of association of genes on a chromosome, right? Now the term recombination was to describe the generation of non-parental gene combination. So we'll see basically with this structure. If you see here, you have a yellow white with wild type gene, right? And the other crosses white miniature with wild type. Now, if you see here in the F1 generation, basically you'll find two combinations. Now, here the wild type gene has come in the first combination, so this is wild type, and in the other one, obviously, yellow white will be dominant. Here as well, you'll see that wild type is dominant here, and you have the white miniature one here. Now, if you see the F2 generation, what you will find is that there's one combination of wild type, then there's one white, just white. There's just one yellow, and then there's just one yellow white, right? So you'll see that the parental type is higher here. Now, why that is the case? Again, I'll repeat the same line. It is because the two genes which are present here are on the same chromosome. These two genes are present on the same chromosome. While in the cross B, what we are seeing that the two genes are not present on the same chromosome, and that is why the parental type, if you see the parental type percentage, that is less, and the recombinant type, which is basically the non-parental genes, is more. So this basically was the third variation which was given by Morgan, which is linkage and recombination. So, guys, with this, we have finished the dihybrid cross and its variation as well. It is a bit complex, but I hope you would have understood it. This is just what we have to study as per NCERT syllabus. We will see more once we study the other books. So, if if you have any doubt related to this video, please do not hesitate in asking me. You can simply drop in a comment on the video, or you can also text me on the WhatsApp number that I have given in the description of the video, and I'll clear your doubts. And if you did understood the video very well, please do not forget to like it, and you can also thank me once over the comment. Thanks, guys. Bye bye.